Okay. Well, I think these few students who are waiting to come in, I think they'll figure it out. Seems like they have no connection. Okay, so this is your microphone. I'm talking about the lab quiz.
The lab quiz wasn't too bad for me, but
Okay,
the
cool.
exam was
All right. I know it's
horrible.
it's a larger quiz, so sometimes students struggle. That's pretty much how most of the lab quizzes are going to be. Some of them are going to be probably closer to 10 points versus 20 points. I know there's like 20 points quizzes. Those are the ones that can't be dropped. So this is the one you want to try to do well on because we can't drop it. That's important. So now let's see. How's lecture going? What are we doing? I think, are we doing atomic theory in lecture?
I just took my first exam horribly.
Do we not attend lecture? I'm assuming. Does nobody know what's going on in lecture?
Did you hear me?
Okay. Kelsey, do you know what's going on in lecture? That's all right. I mean, we all have to. I had to just get out of the shower and shave real quick. I looked a little ratty this morning. So we doing atomic theory? Is that right? I mean, am I wrong on that? Can we talk about something like that? Ionic bonds, polar bonds, covalent bonds, protons, neutrons, electrons. Are we okay? That's usually a hard chapter. Chapter 2 is pretty like one of the hardest chapters, in my opinion, just because it takes you back to Gen Chem. Okay, what's chapter 3? Maybe we haven't done chapter 3 yet. Okay.
It's the cellular form and function. It's the cellular form and function. Chapter 3.
Okay.
Yeah, chapter 2
It's all
was
right. It's no
hard.
big deal.
Can you hear me?
Because I don't know what chapter 3 is. If we're okay on lecture material, this will probably be a relatively short spotlight.
Can you hear me?
No, Relder, I can't hear you. Okay. Okay. I'm glad, Bethany. I'm glad you can hear me. So is there topics that we want to talk about in chapter 2? I'm sure I knew chapter 2 was hard. I had a feeling it would be. Do we want to cover some topics on chapter 2 that maybe I can help clear up? And Relder, if your microphone's not working, you can always chat me a topic. Kelsey, what do you want to talk about? You guys, polypeptides and saturated molecules. Is that right, Bethany? Okay. All right. Let's talk. Thank you. I'm glad everybody brought something up so we can get something out of this. It's horrible when I have to sit here and stare at a chat and nobody has anything to say. I know it's 8 a.m. on a cold morning, but thanks for being here. Let's talk about polypeptides. Polypeptides are basically another fancy term for what a protein is. Okay, a protein. This is biochemistry. This is my stuff. Proteins are made up of polypeptides. Okay, polypeptides, and you guys feel free to chat something in here and direct my conversation. Okay, if something doesn't make sense, either speak up or chat me something, and you can drive this conversation wherever you want it to go. Okay, but I'm just gonna give you my monologue on polypeptides. But if I'm going down something that it takes you away from this class, you need to redirect me back in. Okay, because I'm a lab instructor and I don't have the PowerPoints in front of me. But polypeptides are the fancy scientific word for proteins. Okay, we have proteins. We have thousands, if not millions, of different types of proteins in our body. All right, and what happens is that when we have DNA, right? We have DNA in our cells, and what happens is that these DNA will get coded into mRNA, which is this messenger RNA, and then this messenger RNA, depending on what different nucleotides it has on it, the next step, the next step from the mRNA, which is transcription, the next step is called translation, and that is when we take the mRNA and we make amino acids. Okay, and what happens is pretty much like a bunch of little amino acids that are connected, and each of these amino acids, when they're connected, they create what's called a polypeptide. Poly means that's the prefix for many. Okay, so poly, mono means one, di means two, poly means many. All right, so we're talking about many. Peptide, that's just a fancy word for protein. Okay, so we have many protein. Okay, that's just many amino acids that make up a protein. And so we have amino acids, which are like the building blocks of the polypeptide. Okay, we have nucleotides that are the building block of DNA. Is this making sense? Do we need to go more in depth? Okay. All right. Do we need to dive further in? I know we're going to do saturation next. All right, nucleotides. All right, let's do nucleotides. Nucleotides there's 4 different types in DNA, right? There's adenine, thymine, cytosine, and guanine. All right, ATGC. Now, they pair. When we talk about DNA in us, we are what they call a double helix, right? So we have 2 DNA strands that are met on top of each other that have what we call hydrogen bonds between them. Okay, so we have you probably remember hydrogen bonds from chapter 2, and adenine. It's not adenosine, it's adenine. Adenosine's ATP. Adenine will hydrogen bond only with thymine. So we have I'm going to type this in the chat here. A bonds to T, and then G bonds to C. So that's adenine only bonds to thymine, and then guanosine only bonds to cytosine. Now, did we talk about, this is DNA, Bethany, correct? This is DNA, only DNA. We'll talk about RNA too if you want. Now, did we talk about in lecture about the different hydrogen bonds? GC, guanosine and cytosine, actually have 3 hydrogen bonds between them. All right, so imagine that equal sign between GC and just add another line under it, because each of those 2, that equal sign demonstrates 2 hydrogen bonds, right? GC is stronger, all right? It's a lot stronger. It has an additional hydrogen bond between those 2 nucleotides. It makes the DNA a lot stronger and harder to split apart when we undergo replication. And then adenine and thymine only have 2. So there may be questions depending on the test of, if they were to give you what a DNA strand looks like, they may ask you what the complementary strand would be. Okay. Now, when DNA is made, it's made from the 5' to the 3' end. Okay. So you have to orient yourself when you're looking at a bunch of nucleotides The left-hand side will more than likely be the 5' end. They'll probably write on the question 5', and on the right-hand side, it would be like the 3' end. Okay. Now, if the question asks you what's the complementary strand, you have to remember we're making DNA always from the 5' end. So if you want to make a complementary strand, more than likely the complementary strand is going to be the 5' end on the right-hand side. Okay, not the left-hand side, because it's opposite. We cannot make DNA. The polymerase cannot go from 3 to 5. It has to go from 5 to 3. So it's only one direction, right? It's not this direction, it's this direction. Does that make sense, or does that complicate things? I don't know how in depth we're going here in lecture. Give me something, and then we'll move on to RNA. All right, thank you, Tristan. Okay. I'm gonna, yeah, I had a feeling, Relder. I had a feeling. All right, let me get a piece of paper, and we're gonna draw this out. Give me a minute. Okay, good. This is what these spotlights are used for. So when we get interaction, we can really get on a cool topic, and like we can clear out some cobwebs, right? Okay. That's okay, Anna. You'll get it from me, and then likely you'll get it from him. So when he goes over it, you already have one pass at this material, and then you can understand it when he goes over it. It's a lot easier. All right, I got a new kit, got some new pens out. We're gonna have some fun with this. All right, so this is my, yeah. I have my outside cat hollering at me through the window. Hold on one second. I'm gonna write this out. Come on. There. Sorry, if I would have left her there, she would have cried the whole time. All right, so I'm going to do adenine, thymine, guanosine, cytosine, and this is the 3' end. Yeah, and Anna, just you can record them. I got a YouTube channel now for my students in lab. They really wanted it. So if you all want to watch this again, you can look at it through my YouTube channel. I think people ask me what it is. Just Google my last name, put like Bio137, and I'm eventually going to have this up, and you guys can take a look at it. All right, so this is the parent strand. Okay, this is the parent DNA strand. Okay, can everybody see that very well? That is from 5' to 3'. Okay, cool. All right, so that's the parent strand that we're going to use this from. Eventually, I'll get real good, and I can like copy this and I can write this down, but this is the best I can do right now. So, polymerase. This is DNA Pol. Polymerase, which is right here, that's going to go from 5' to 3'. Okay, that's the only way it can go. That's the parent strand. It is going to make new DNA. So all of a sudden we're going to have A pair with T, T pairs with A, G pairs with C, and then C pairs with G. That green stuff is the new strand. Okay, that's the new strand. Now, it's opposite. So over here, this is the 5' end, and this is the 3' end. Do you see those numbers in green there? that's the new strand. Okay. So we have 5 prime. Let me see if I can get this right. 5 prime to 3 prime. That's the parent strand. Polymerase is only going to be able to go this way from 5 prime to 3 prime. Yes. Oh yeah, Bethany, if you want to talk about a child, yes, this happens all the time. This is cells growing. This is called replication. And you're going to get into this. You're going to learn this, and then you're going to learn what mRNA is and proteins. So this for both. Yes. Yeah. And this is your epithelial cells, your skin cells, your hair cells. This is everything. And then eventually, for the new strand, it's going to be from 5 prime to 3 prime, because this is going to split. Eventually, the new strand is going to become the old strand. Okay, this is like mitosis, right? Remember mitosis? We've probably done this in lecture or lab and lecture. When we split the DNA and we make new cells, all of a sudden the new strand is going to become the old strand. So then polymerase is going to jump on the new strand and it's going to create another DNA strand. So cancer is a big deal. So cancer basically deals with when we have, we have a cell cycle. Remember, we have a cell cycle in mitosis. Mitosis is basically what happens when we recreate DNA and we split cells, and we have this cell cycle, different stages. We have G1, S, G2, and the M phase. These are different phases of the cell cycle. When the cell enters these different phases of replication, it checks itself, right? That's what it's normally supposed to happen. It checks itself and makes sure that it has the right components to make DNA, because DNA is a very important process, right? You don't want to screw up DNA replication, because if you do, something bad can happen, i.e. cancer. However, when you have this tumor or this proliferation, what happens is that the cell cycle, these phases, all of a sudden there's no check limits. It just replicates and replicates until it cannot replicate anymore. Okay, this would be for like leukemia, which is white blood cells in your body that just keep on replicating in the bone marrow. Lymphoma is a tumor of B cells, basically, that come together that will take a lymph node and blow it up. And this is kind of the beginning of how cancer gets started. Is that the cell cycle pretty much goes unchecked, and it just continues to replicate. Your body doesn't want to sit there and continue to replicate everything. Okay, if it has enough of something, it's going to stop it. It's going to halt it. We just don't sit there and crank out cells. It will regenerate cells as it is needed, because we really need to budget our energy. So what happened to the checking process? Is it deleted from memory? It's a mutation. It's a mutation from the DNA. It all goes back to DNA. It all goes back to genetics. So when I talk about this DNA strand will code for mRNA, which is RNA, right? Well, and Bethany, I haven't forgot about your question. We're going to go to RNA before we're done here. And then the RNA is going to make protein. So this protein has a job. Every protein in your body has a job. And it can be either to help out a system or to suppress a system. Anna, if you really want to get into a particular protein that is very important in life, you can Google P53. P53 is a very important protein called the tumor suppressor protein. All right. It's also made from the P53 gene. Yeah, P53. That's correct. And you can Wikipedia it, and I'm sure it was a huge discovery a number of years ago, because people who have a lack of P53 are more likely to develop cancer over time. And what P53 normally does is that it keeps this cell cycle in check. When this gene that codes for P53 protein, because you have to remember, the protein's the one that carries out the jobs. The genetics is just there as like the recipe, right? So let's say we're cooking chili, right? Because it's cold out. Well, we have a recipe that tells us how to make this awesome chili. And then all of a sudden we put all these components together, and then, you know, what makes the chili good is that we have hot beans in it. And hot beans is what really makes the chili good. Well, in this case, the hot beans are the protein. The protein's the one that carries out the job, that makes things good or bad. And in this case, P53 is a suppressor. So you have to have P53 around to suppress tumorigenesis, which is the making of a tumor or the production of cancer. When this gene is knocked out, or this gene is no longer effective, we are no longer making P53. And in that case, there's a large chance later on in life that we can develop some kind of cancer. Does that make sense? Yeah. Okay. Yeah, that's a lot. Yeah, you guys are hitting in my wheelhouse here, so we're hammering down. No, Dr. McCain, Tristan does these. It's her and myself that do these. Yeah. Tristan, all the classes are the same. Who do you have for lab? Or who do you have for lecture? Let's do both. But everybody's supposed to be on this. Everybody's supposed to be on the same page. Okay. I share a Blackboard shell with Karen Drake and Dr. McCain. Yes, Dr. McCain's the course director. Okay, so let's talk about RNA real quick. I know Bethany mentioned that before. RNA is basically the same thing as DNA, but there's one change, and this will be test questions and quiz questions, probably sometime in lecture. You do not have thymine in RNA. That's the T, right? Remember the T right here? See if I can see that. That's a T. Remember the T? So if you don't see the T and you see a U, that is uracil. All right, and that is a nucleotide that is in RNA only. Thymine does not make it to RNA. It will still, so if we are replicating DNA and we see a thymine, that thymine automatically just gets changed to uracil. Okay, and so that means that adenosine or adenine will hydrogen bond with cell in RNA. And that's pretty much RNA. Does anybody know why we don't use RNA as like a genetic source? You know, DNA is the one that pretty much makes us who we are, okay? You get it from your mother and you get it from your father, and DNA is pretty much all the genetic memory material. If you have children, how they look, their noses, their facial features, like this is the Mattmiller nose, okay? If you were to run into any of my family members, we all have the same nose, because the genetics of our family make this kind of nose. If you were to see my uncles and maybe some of my older cousins, you would see that they're half damn bald. And now that I've been on my YouTube, yeah, okay, see, Kelsey, that's right. So when I watch myself, which is so weird, I don't do this a lot because it's odd, because I'm new to YouTube, I see how bad my receding hairline is. And when I see my cousins and older uncles, I'm like, oh my gosh, I'm looking just like them. And that's because we tend to have the same genetics. Okay, there's a little bit of variation, but everybody seems to have the same features, and it's all because they have similar genetics. And the point I'm trying to make here is that RNA is just a messenger. It's just like the stepping stone to make proteins. We do not store any information in RNA. And that's because it's far less stable, okay? RNA just gets like wrecked really quickly. Messenger RNA does not last very long at all. DNA can last for a while. You know, crime scenes, we see CSI people, DNA, like they could pick up DNA from like years ago. Like if they went to go find some prehistoric person or Neanderthal they pulled out of England back in 1200 BC or 6000 BC, whatever, they can likely recover some form of DNA from that Neanderthal. What is it? What am I looking for? I'm looking for cave people or whatever it's called. I cannot think of the word. And DNA basically is the point. The reason why we store information in DNA is because it's very, very stable. And that's because it has hydrogen bonds. It goes back to this concept we first introduced, the hydrogen bonds between A and T and G and C. Does that make sense? I don't want to eat up too much time. Okay. All right. Are we soft on anything? Do we need to move on to another topic? This is enjoyable to me. We're actually talking about something interesting instead of, okay. All right. Bethany can move on. That was okay. I think we answered your question. Tristan, you're good. Okay. Relator, I guess you're okay too. Okay. Now, Bethany or somebody talked about saturation, lipids. Okay. All right. So saccharides, I'm not sure. I mean, that's not exactly a biomolecule. You might have been. Okay, good, Kelsey. You're good. All right. Well, I got this on recording, so if your hand's cramping up, you can always check this out on YouTube. I'm probably going to get it up sometime this morning. Let's see. What was it? Was it lipids, Bethany? You talked about. I'm trying to go through this chat here and lipids, saccharides. Can you elaborate on that? Okay, carbs and lipids. All right. So these are biomolecules. You're talking about biomolecules. All right. So let's start with carbohydrates. Carbohydrates is another, it's how we get energy, right? We can get energy from fats or lipids. We can get energy from carbohydrates. We can get energy from proteins. However, each of these biomolecules have different percentages of energy. Do you know what I mean by energy? What is the energy? What is the energy source of this? Yeah, that's right, Anna. Very good. It's ATP. ATP. So, and when I'm talking about ATP, I'm talking about calorie, right? Remember, everybody's heard of calories, right? How many calories? That is basically the unit. Are carbs and saccharides the same? Yes, yes. A polysaccharide is the same thing as a sugar or carbohydrate. Yeah. Pretty much. So the reason why we humans in general prefer sugar is because sugar or carbohydrates pack a lot of ATP. All right, and we like that because from an evolution standpoint, way back in the day, we never had carbohydrates. You know, it was always meats and proteins, right? And maybe some plant-based food that people would cook up, but they never really had a lot of like bread or starch, you know, foods with high carbohydrates. So if you think about it, that's why people, you know, and that's why people, you know, the obesity rate is sky high is because our bodies are not used to having this much carbohydrate intake. So that's why we store more sugars than what we consume, and that gets transferred into fat. Okay, and that's why, you know, obesity is a problem these days. So carbohydrates basically have anywhere from like I can't remember what it is. It's 9 or 12 ATP units per calorie. So I'm not trying to blow your mind up. What I'm trying to say is that calories are the measure of energy, right? Well, how many calories does this have? You know, and that says basically what that means is how much energy does this Coke have, or how much sugar? And when we're thinking about that, we're talking about sugar, which is packed with glucose and stuff like that. So when we eat fruit, yes, fruit has natural carbohydrates in it, or what we call glucose, correct? Or it's called sucrose or fructose or maltose or lactose. Those, and like I said, I'm not trying to blow your mind. I'm just trying to show some examples here. Those are all different types of sugars, okay? Lactose intolerant people who can't eat lactose, the reason why they can't is that some people lack the protein to break down lactose. All right, lactose is what we call a polysaccharide. All right, so, well, it's actually a disaccharide, but we'll just call it a polysaccharide. For us to get energy, we have to break polysaccharides down into monosaccharides, one sugar. You know what that one sugar is? Anna just said it. It's glucose. We have to get sugars in the glucose form, mainly to utilize them in metabolism. So let's say you go eat like this morning. I was up early. I went to go get donuts. Okay, I said I'm going to go get some donuts. I'm craving these donuts. I'm going to go get them. Now I only limited myself to one donut. So this is a bunch of sugar, high, like bread is like a lot of polysaccharide, glucose. So what happens is when I eat this sugar, my body will break it down into one single sugar, which is glucose. And that's how we can extract ATP out of that glucose. We have to break it down. And we can break down lipids, and we can break down fats, and we can break down proteins. And all that yields some kind of ATP. But it's just a lot. Our bodies tend to crave the sugar and the sweetness just because it has a lot more energy to it. And we are trained to get as much ATP out of food as possible. And that's just an evolutionary standpoint. And sometimes that's why we overeat our calorie count, 2,000 calories per day. And, obesity, the more calories we take in, and if we're not very active, then those calories get turned into fat. Are you guys cool with that? Okay, kind of making sense. All right. Okay. So let's talk about lipids. Now, lipids are interesting. Lipids, let's talk about like biomolecules of lipids. So when we go out and we go to the pizza parlor and we get a pizza, we're basically getting a bunch of cheese, bread, and sauce. The bread obviously is going to be glucose and polysaccharides. The cheese, on the other hand, is going to be lipids or fats, right? And there's good and bad. There's good fats and bad fats. And lipids basically are what you've probably heard is a nonpolar compound. Okay, a nonpolar compound, which means that they don't bind to water. If you were to put a bunch of lipids, like lipids and water together, like oil and water or something like that, you'd see that they'd separate. And that's because lipids bind lipids. They don't bind water. Some of them do, but lipids themselves do not. If you think of soaps are kind of like your hand soap or your body wash soap. The way soaps can clean your skin is that lipids tend to bind together and to create these little circles called micelles. And the interesting thing is that you have like these little balls of lipids that bind each other, and as they run across the surface of your skin, they can attract dirt and grime and other stuff, proteins on your body, and pull them off, extract them. And that's kind of what soap is. But to get back to biomolecules, this is what we call triglycerides. Have we ever heard of triglycerides? If we ever heard of HDL or LDL, that is just fancy terms for a type of lipid. LDL, which is something that you would probably have a problem with if you have a poor diet, you eat a bunch of pizza or cheeseburgers or fries, you would have a high LDL content or bad cholesterol. It's cholesterol. And that's what essentially in your body, when you eat a bunch of lipids, how your body breaks it down is that it breaks it down with cholesterol. Yeah, that's right, Bethany. And this is how, this is when you eat a piece of pizza, we want to extract some of the lipids out of there and either discard it through your excrement, or you will keep it, right? And use it for fat, use it for cell membranes, all kinds of different things that lipids can do. And how we do this is that it will use cholesterol. There's different forms of cholesterol. There's LDL, like Bethany said, and then there's also good cholesterol, which is HDL. And for instance, I try to take a fish oil every day because fish oil or salmon, fish have high levels of HDL, and sometimes males tend to struggle with HDL levels. And I've noticed that I've always been on the lower end of HDL, and that irritates me. So I try to take a fish oil. It's good for your heart health and stuff like that. However, so if you were to have a high cholesterol or high LDL because you have a poor diet, low density protein, high density lipo. Excuse me, I'm forgetting the lipoprotein part. No problem. And you can Google that if you'd like and read into it. So if you have a high lipid diet, okay, yeah, Anna, it can. Well, for females like estrogen, progesterone, there's a few others, and maybe those are two big ones. Those are what we call lipid-based hormones. Okay, they are, and those are crucial in females and the menstrual cycle, and especially estrogen and progesterone. And there's the follicle stimulating hormone and luteinizing hormone are water soluble. Those are not lipid-based hormones, but like cholesterol-based. They're basically cholesterol-based. And so if you were to eat a bunch of pizza, that means your body's got this huge influx of lipids. So what it does is that it has to ramp up its LDL protein, right? We talked about protein before. Well, it has an LDL gene that makes the LDL mRNA, and that LDL mRNA will make the LDL protein. And that will help absorb these lipids and either put it through your excrement, or they'll retain it. Hold on, Bethany. These lipid base. Not as there that could go a lot of different ways. Bethany. I think progesterone, when you think of that, is depending whether you're pregnant or not pregnant. I believe progesterone will help sustain the pregnant. Well, HCG basically helps sustain the pregnancy. Progesterone will help keep the vaginal mucus layers thick throughout the pregnancy. Okay. There was a time where I was really good on reproductive. It does. I have PCOS. Okay. Yes, PCOS is something very common in women, and sometimes estrogen and progesterone can get involved. I think, we've all heard of this Plan B pill. Plan B is basically just a huge dose of progesterone that pretty much sheds the vaginal layers. Hold on, Tristan. So guys who want to take the roids, right? You're talking about roids, steroids as we call them. So that is basically that is a version of testosterone. Okay, testosterone and acetate, and basically it's cooked up testosterone in the lab that you will pull up through a needle and shoot it into your ass, right? Because if you think about it, you are pretty much injecting testosterone into the fat of your bottom. Okay, I should be polite. I'm being recorded. Into your bottom. So what I'm trying to say is that what this testosterone does, testosterone carries out a lot of different stuff, right? It will boost up it'll boost your LDL up, which is not good, which could give you heart issues over time. And what I mean by that, I mean like a heart attack, because it messes with the fat deposits in your arteries. But testosterone will boost up your muscles and make them big and buff. If you take it for too long, your body relies on this testosterone, and your scrotum will shrink. Okay, because basically it's getting all this outside testosterone. So why do we need to make inside, why does your body need to make normal testosterone if you're shooting up with outside testosterone? And basically your whole, what we call native testosterone, your native self testosterone will like plummet. Now, estrogen will turn into testosterone. It's kind of interesting. If you have too much, oh, okay. Yes, sorry, I'm trying to think about this because I'm trying to think about if testosterone or estrogen. Testosterone will turn, if you have too much testosterone, it'll actually turn into estrogen. Okay, I think my audio's on. I want to say testosterone. Okay, I'll type this out. That's not how you spell testosterone. Testosterone, too much. Let me type this out so you can understand. Too much testosterone can turn into estrogen. All right, and if you're a male, if you have too much estrogen running around in your body, you can actually grow male breasts. All right, now that won't happen normally. That only happens when you take steroids, and there's a word that I'm not going to use, but it's called. It starts with a B, and then it's kind of like the slang term for breasts. And that's what you try to avoid if you're taking steroids, because if you take too much testosterone, your body will convert that into estrogen. And males do actually have a little bit of estrogen. Estrogen, quite frankly, plays a lot of different roles in just reproductive. In children, estrogen will help in the growth plates of children and how they grow. Okay, like these kids that are basketball players, they have a lot of estrogen in their growth plates, and the amount of estrogen in the growth plates help the growth plates grow, and that's how they can become so tall. So it's kind of misleading. Does it go the other way for women? Too much? I don't think so, Bethany, because there is a protein enzyme that, it's all going back to proteins. There's a protein that is proteins and enzymes are the same thing. Okay. All it is that an enzyme carries out a reaction. All right. It's like I said, like that P53, you can also say that that's an enzyme. So there is an enzyme called an aromatase, right? And we don't want to dive too much into this, but if you have this aromatase, will take the testosterone and convert it into estrogen. All right. And I think, Bethany, that at one point when females are kind of going through the puberty stage, that they will make testosterone, and then it quickly gets switched over into estrogen because females have a higher level of this aromatase. Okay. Now, if you all want to dive into what happens when we have, okay, I'm not sure, Tristan, how to fix that. If you pair it with headphones, it will kick me out and I still can't hear. Okay. I'm not sure. You might want to just try to use your computer. You may want to email me if you have problems there. When you have issues with babies with what we call ambiguous genitalia, this is what happens when we don't know whether they're a boy or a girl. And they have to do certain genetic studies to determine whether they have what the sex hormone is, what the sex gene is. If the male sex gene, they have, obviously they're male, and sometimes they'll have like the X or Y chromosome. Sometimes they'll have two X's and a Y. We're either a Y or we're X. And sometimes these people, the body can't, something's happened in the development. And it's like Klinefelter's is a Klinefelter syndrome, Turner syndrome. Those are the big ones where you have, we've also heard of these athletes that this is not transitioning. because this is something that they're born with. And they may help it and they may take testosterone shots, but this is a genetic based issue. And sometimes you can have more estrogen and testosterone kind of mixed in, and you have to go see a endocrinologist to figure things out. So that's a whole other ballgame there. That's a lot more in depth. Okay, a lot of questions. Thanks, guys. We kind of started off slow, but this has been a nice conversation. Bethany, I just saw your facial hair. That can be a lot of different things. Estrogen and facial hair, yeah, sometimes like if you have too much facial hair, that may be from just circulating testosterone in your body. That's like secondary sex features. And obviously estrogen has secondary sex features in females in the groin area, and but everybody's going to have hair underneath their armpits. Everybody's going to have hairy legs. That's just genetics. And I'm not sure if you can relate that back to estrogen or not. Okay, sure, Anna. ATP. No, that's fine. Well, if there's questions that come up, Bethany, let me know and I'll try to answer them. Anna, let's talk about ATP. ATP. This is going to be kind of like a quick down and dirty explanation because we could probably spend a lot of time on metabolism. It's probably one of the more complicated concepts, but it's also one of the more cooler ones. If we remember, we talked about these polysaccharides. I just ate that donut, right? If we eat that donut, my body's going to break this down. It's also going to cause my blood sugar to go up. And what happens is eventually we get glucose. And glucose is a monosaccharide that we will put into my body, and it will undergo metabolism. Now, there's different stages of metabolism. Let's talk about this. We got 10 minutes. The first stage of metabolism is glycolysis. Okay, glucose gets broken down into two molecules called pyruvates. Okay, just bear with me here if I introduce some terms to you. If you've never heard of them before, then you're likely don't have to know them. But if you want to hold on me and ask me more questions about it, let me know. Glucose will get broken down into pyruvate. Pyruvate will go into another phase of metabolism called the Krebs cycle. It's also called the citric acid cycle, and maybe something else, or CTA cycle. I've known it as the CTA cycle. Other people call it the Krebs cycle. It's the same thing. And what happens is that pyruvate will go through these different stages, and it extracts electrons out of these molecules. And what happens is these electrons are used to do something else. And we pull out all these electrons from glucose because it's like going through the car wash or something. Or it's like going through like an assembly line, but we're taking, instead of adding stuff, we're taking stuff, and we're taking electrons, right? So at the last stage, it's called oxidative phosphorylation, and that's when yeah, Bethany, let's get to that. I think you're right, but let me finish my concept here. When you go into oxidative phosphorylation, you have ADP, adenosine diphosphate, not adenosine triphosphate. Adenosine triphosphate is ATP. That's the energy that we want. ADP basically is like a body with, let's just say, this is kind of a crude example, but this is the best thing I got because we're running out of time. Think of it as like we're missing something. Think of it as like a body without a head. Okay, I hate that I'm going to use this expression. So but when we put, it's like it's missing its important piece. All right, and let's call that the head, because this is what I see in my screen right now. If you have a body that is headless, you have ADP. these electrons are used to power a phosphate onto that ADP. That's like putting a head on me, and all of a sudden now I'm adenosine triphosphate. Okay, because all those electrons that we used build up enough energy to place a phosphate on the ADP to make it ATP. And that's the only type of energy molecule that we can use. All right, and one glucose will yield 36 ATPs. Does that make sense? That's how we get, yeah. More, yeah, I think that's net, yes, that's net, that is net ATP, okay? It's actually, we'll make 4, but we actually use 2. So yes, we net 2 ATP in glycolysis. Krebs cycle, we'll have to double check on that. It may be more, maybe 6. Don't quote me on that, maybe 6. And then oxidative phosphorylation does the electron part, but you can Google that if you can check. Maybe Relder maybe says you're right. I'm not sure, but that's the same concept. Yes. So does that help answer your question there? That's a lot of different stuff. Okay. All right. We just want to make sure we take ADP because that's like the skeleton. And then once we put a head on it, then it's like it's good to go, and the body can use it. All right, so Bethany, you talked about acids and pH. That's interesting too. We could do another hour of that. That is very important in the clinical world. Acid and base balances, and we can go all into depth with that. What happens when you work out? Why you're sore? What is lactic acid? What does lactic acid do? And basically, when you have, let's say you go out for a run, you haven't worked out in a long time. You build up a bunch of lactic acid because your muscles are not used to having all this exercise. And this is what glycolysis does, actually. Glycolysis is metabolism without oxygen. And it happens when you are out of shape. If your heart rate's running, you're sucking in all this air, you're not getting enough oxygen to your muscles. And your muscles are doing all this work, and they need oxygen, and they don't have the oxygen. So what happens is that they make this lactic acid because they don't have the oxygen to get rid of it through glycolysis. And what happens is that all this acid builds up, causes your muscles to get sore, and that's why your kidneys have to work out the lactic acid over a 2 to 3 or a 1 to 2 day period. And so yes, you have more acid, which means you have more hydrogen atoms, or what we call protons. Okay, protons are also hydrogen atoms. They build up and they lower your blood pH, and that's how you have more of an acidic feel to it. Also, your muscles become sore. It releases water, and more basic, the more. If something is more basic, Bethany, that means they have more hydroxyls, or OH. Hydroxyls would be this. Yeah, there you go. I'm trying to spell it out, and I'm blanking. Yeah, or OH. Okay. So yes, that is more hydroxyls. Yeah, they can, because water is H2O and you already have OH, right? So if you add a proton, you get H2O. Okay? Does that kind of make sense? More basic, more hydrogen interacts and mixes with water. Yeah. Yes. That is basically what happens when you, if next time y'all, hey, we got 2 minutes left. I'm doing another one on Sunday. You all see them on there. If you want to come in and bring this up, we can talk an hour about this. If you want to get into what happens when we do carbon dioxide, this is kind of what Bethany's talking about. What happens when we have bicarb and lactic acid, and what happens when we get a hangover, or what happens when we have when we work out too hard. I think it's 7 to 8, Relder, 7 to 8, and you may want to sign up, because there's only like a limit on these. But if you can't get in on Sunday, we're doing another one on Monday. Doing another one on Monday and another one on Wednesday. Oh, excuse me, no. Monday 7 to 8, Wednesday 8 to 9, and then Friday 8 to 9. So let me write this down. Sunday 7 to 8 p.m. Monday is 8:45 to 9:45 p.m. I was skipping that. Wednesday and Friday 8 a.m. 8 a.m. So those are my spotlight. Those are my spotlight times. If you guys want to jump in another one, we could make an hour go by really fast if we talk about interesting stuff like this. And hopefully, the way I present it, you'll kind of can relate to it, and it'll help you remember. So, is there any other quick questions by chance? Oh, let's do the. While you all are thinking of questions, let me do the attendance form real quick. Thanks, Relder. I had to brush off some cobwebs there, but I enjoyed talking biochemistry. Biochemistry is pretty much the marriage of chemistry and biology. Oh, good, Anna. I'm glad. Now, you guys, if you've done this already, or if this is your first one, do the attendance form, and I'm going to sit here and wait, and then let me know when you're done. Just put Mr. Mattmiller, Friday morning. Yeah, yes, Tristan, it's the attendance. Okay, Anna, thank you. You're good to go. Have a good weekend. All right, Bethany, sounds good. Stop in next time or catch me next week or sometime. We'll hit this stuff again. Have a good weekend. Okay. Thanks, Kelsey. Try to stay warm out there. It's cold. Let me text Tristan and let him know. Yes, I think so, Relder. So Tristan, I guess you can't hear me because your microphone's off or not working. It may be something with your speakers. I want to throw this up on YouTube, and hopefully you can watch it. Okay. All right. See you.